Welcome. This webinar is brought to you by the New Hampshire Training Institute on Addictive Disorders. You can access the recording of this webinar on our website at www.nhadaca.org within our webinar library. After completing this webinar, please complete the quiz and evaluation if you're interested in receiving a certificate. You can find the quiz and evaluation on our website next to the Watch Now button you selected to view this webinar. If you're a member of NHADAC or a NADAC, the certificate for this webinar is free. If you are not a member, the fee is $15. You will receive a certificate worth 1.5 continuing education credits once you have completed and passed the quiz and paid for the webinar if applicable. Through the New Hampshire Training Institute on Addictive Disorders, this 1.5-hour event is pre-approved by the New Hampshire Board of Licensing for alcohol and other drug use professionals, as well as NBCC. If you have any trouble viewing this webinar, have audio problems, or have any technical questions, please contact us at 603-225-7060. Please email any questions regarding the material you see today to training@nhadaca.org.
I'm very, very excited to be able to talk about a topic I've been researching on, and it's really dear to me, and I'm really passionate about it. Just a quick background about me. I have been teaching for 20 years anatomy and physiology in college settings. So I'm an assistant professor right now at Franklin Pierce University in New Hampshire. I also have a research lab on campus, and we started a research project on exactly the effects of opioids on the human brain. That is exactly the topic of my research. And I'm going to have a chance to share some of that with you towards the end of the presentation, as I'm going to go through some, background knowledge and then take you into more of the details of what we do in a research lab to really try to understand what's going on. What else can I say? I really love what I do, and if you feel you have questions about anything that I'm talking about, you can actually stop me. I can repeat something, clarify something. If you have questions, you can unmute yourself and just talk. Or if you prefer to write it in the chat log, I'll be notified of any particular question that need to be asked, and we'll take, breaks, question breaks, and things like that. What else? Let's see what the objectives are, and then we're going to go through this together. Okay, so these are the learning objectives for this course. Describe the normal physiology of the reward system in the brain. So trying to explain to you how it all works just in a normal setting when you feel rewarded, and explain what happens when opioids come into the picture and how they're actually changing that reward pathway and tweaking that reward circuit. So I'm going to explain to you how they do that at the physiological level. And then through the discussion of my research, I'm going to take you into a more of a view of how these opioids have a long-lasting effects on overall brain function. And I can explain to you some of the thoughts that we have right now in research about why is it that people relapse so often, and this grip. That's why I added that in the title. This grip that it has on the circuitry in the brain. So I always like to start with just some history, just so you have an idea of the opioids and the background, the historical backgrounds. I think it's really important, and this is a little bit of the outline that I'm going to go through. Just give you an idea of where we're at in the lecture. Okay. So something that a lot of people confuse are the terms opiates and opioids, and or use them interchangeably without really realizing that opiates are the analgesic compounds that are actually naturally found in the seed pods of opium poppy plants. So this is where you can extract these natural compounds, and the examples are morphine and codeine. Those are actually naturally found in those seeds of these flowers. Now, we use the term opioids to describe all the medications that reduce the intensity of pain signals reaching the brain. So reducing intensity of pain signals, that's basically the definition of an analgesic. So analgesic is like we saw in the previous slide. These are analgesic compounds. That means they reduce pain. And that's really why we use them in a hospital setting, in a medical setting. We use them because they reduce pain, and they're very strong. So it's helpful for a patient that's in a lot of pain. Now, they also have other effects, and that's where some of the problems are. And we're going to talk about that. So when we use the term opioids, this is actually basically includes all of them, the natural opiates and the synthetic counterpart. So synthetic, that means that there's a chemist in a lab that has taken the natural molecule that's naturally extracted from the plant itself and tweaked it chemically. And literally heroin just basically have an acetyl group on it, on the morphine molecule, and that's it. It becomes heroin. And heroin is a synthetic counterpart of morphine. So in that umbrella of opioids, you have all of that, all the natural ones, and the natural opiates, and the synthetic ones that were produced in a laboratory. So some of these are examples of hydrocodone, fentanyl, heroin, even though it's not produced commercially anymore, but those are synthetic. So how did we end up with really a synthetic compound like heroin? Actually, this was produced in a lab chemically by Dr. Alder Wright back in the 1874, and really eventually got banned in the 1920s in the United States, because we discovered that this was a really addictive molecule. And really at the time, it was really a quest to find a non-addictive form of morphine, because at the time in the 1800s, morphine, the natural opiate, morphine was used as an analgesic and was very helpful, but at the time we knew that morphine was a little bit addictive, so we're trying to find a better version of morphine. And Dr. Wright came up with heroin. And for a while, this was like the miracle drug that really helped and soothe a lot of the pains. And these are actual advertisements from the times where it was used as to suppress cough. It was basically sold in pharmacies. It's kind of like really incredible when you think about it nowadays. So really all he did is add an acetyl group, an additional acetyl group to morphine, and it became heroin. So of course this takes a chemist to do that with the right setting in a lab. But these are acetyl groups, CH3, CH3, CH3. These are compounds, little compounds that we call acetyls, and they're basically plugged onto a molecule, and the molecule now has different properties. Every time you tweak a molecule, even by one atom, it changes properties, and this is how heroin was discovered. And heroin, why it was so popular at the time, it's because it was more potent. It was 2 times more potent than morphine. And because it was such an incredible, potent analgesic, it was named Hero for that purpose. So that's where the prefix hero comes from in heroin, because it was so strong. So we thought at the time that it was like this miracle drug that's going to be better than morphine and not as addictive as morphine. But unfortunately, we realized that wasn't the case. Another drug that was also synthetically produced is fentanyl. And fentanyl is currently used as an analgesic, a very, very strong analgesic in hospital settings to treat, severe pain or chronic pain. Cancer patients are often prescribed fentanyl patches that they use, for very gradual administration on a long, over a long period of time. But it's actually very potent. And just to give you a perspective, I created this little graph here. It's not a graph, it's just my own setup here, showing you, so morphine is here, and heroin is here, and I just told you heroin was 2 times stronger than morphine, and here's fentanyl. So fentanyl is actually 50 times stronger than heroin, and so in consequence, it's also, so that means it's also 100 times stronger than morphine. It's very, very strong. And most of the opioid overdoses are going to be fentanyl overdoses. In New Hampshire, at least. I'll show you some numbers. This is another picture that is very, very visual. It shows you how lethal and dangerous these compounds are. This shows you lethal doses. So with just that amount of fentanyl, you can overdose. And now something even scarier, carfentanil, this is the only amount you need to overdose on it compared to heroin. So these are very, very strong synthetics that can be used in a hospital setting, but it's so dangerous. So it really has to be used in the correct setting. Now there was this story. It's not. I don't know if it's it's something that I had heard about drug dealers and drug dealers switching to fentanyl as opposed to heroin. And you'll see when I show you some statistics from New Hampshire deaths, they're all fentanyl now. But it used to be heroin at some point. And so what drug dealers used to do before, now it's all fentanyl use, is they would add fentanyl in these their different heroin bags, and one of the bags would have, so all of these were heroin bags, and one of the bags would have a fentanyl dose in it. And the people buying the heroin for to ease their pain, because at that point when you're using so you're at a point where you're basically trying to avoid all the withdrawal effects. You're not really trying to get euphoric again. The one person that would end up with a bag of fentanyl without knowing, right, they would overdose and die. And in the mind of the drug dealer, this was like a marketing thing where all of the other users would realize that, wow, this was really some potent batch, and then this dealer might have a really good source of heroin when all along it was just laced with fentanyl. So this is something that it's not, it wasn't published anywhere, but something that I heard drug dealers would do. But now all of the deaths are just fentanyl, basically. So let's just look at some statistics and see where we're at, especially in New Hampshire. This is the data from the conclusion of 2020. So I get this information on a monthly basis from the New Hampshire Medical Examiner, the statistics on drug deaths, overdoses, which drugs, every month. And this is the final count for 2020. And it's better than back in 2017 when it was really at the highest in New Hampshire. We believe that this dip in numbers, even though it's a lower number of overdose deaths, we don't believe there are actually less usage. It's more attributed to the number of saved lives from the use of Narcan. So people overdose, and then they're saved by paramedics or police officers on scene, or now even libraries are allowed to have Narcan in some big cities, Narcan with them. So that's what we think. We think that the dip in the number of deaths is more related to the lives being saved as opposed to less usage. So it's still a problem. All right, so this is the summary of these. Let me move something here. This is a summary of these drug deaths, and I wanted to give you an idea here. So this is for the whole year of 2020. This is the final summary, except that there is additional 14 pending toxicology. So that's how we ended up with the number 411 in the previous slide. So other drugs, we have 56. So that could include so many different other drugs, cocaine and just different other drugs, but all the opioids are all up here in this category. So really making, I believe this came up to be 83% of the final total. 83% is basically because of opioids. And if you look at the different opioids, for example, heroin with no other drugs, there are no more deaths with just heroin overdose. They're pretty much fentanyl or fentanyl and other drugs. That does not include heroin. Here we are. So fentanyl is just really a huge problem now, because you saw how potent it is. And even I just wanted to add something else about the potency of these drugs, the carfentanil being also found on some scenes, death scenes. There are reports of paramedics or police officers that are being intoxicated from just either touching it or particles being in the air. That's how potent it is. So it can affect some people that are on the scene. Okay. So let's move on to talk now about the brain. It's my favorite part, my favorite organ system. I tell my anatomy students that we cover all these different organ systems. Nervous system is my favorite. And my second favorite is the endocrine system. All the hormones that have impact on so many different other systems in the body is really incredible. So we're going to talk about the brain. So I'm not sure of all of your different backgrounds, and if you see me looking sideways, it's because all of your beautiful faces are on my second screen over here. And so yes, hello, I'm happy to have you and to see you. You know, my students, they're not obligated to turn their cameras on, so sometimes it's really weird to have all of these black boxes and to talk by myself. But here I have all of you with me, so I'm very happy. Let's talk about the central nervous system. And because I don't know all of your individual backgrounds, I'm not sure if it is going to be oversimplified, or by the time we reach my research project and the details about my research project, maybe that's going to be a bit too complicated. So there's a little bit of both in this whole presentation, and I hope everybody will get something out of it. So if you feel overwhelmed sometimes, take notes, and I know you'll get the PowerPoint afterwards. take as many notes, draw with me. I'm going to draw a synapse in a minute, and ask questions. So the central nervous system, so let's get back to this. The central nervous system, so the nervous system as a whole is made of two big subregions. The central nervous system, this is really like where all the processing takes place. It's where all the nerve cells, we call them neurons, are located. They're either in the brain or in the spinal cord. All of these nerve cell bodies, the cell itself. And then we're going to see that the cells have really long extensions to communicate with other cells or with organs outside of this nervous system. And that happens via the peripheral nervous system. So all of these nerve fibers that are exiting the brain or exiting the spinal cord, they're all, think of them, I tell my students to think about cables and the cables that are going throughout your house, for example, from the electrical box in your furnace room, utility room. So that electrical box is basically controlling everything, and all the cables that run to through your house are going to help connect that electrical box to the outlets, to the light switches, to all of these things in your house that need electricity and to be turned on or turned off. And that's really how the nervous system works. There's a lot of decision making over here, and more of the we call this the autonomic nervous system, which is all the automatic reactions takes place from in the brainstem, and then in the more of the spinal reflex in the spinal cord. But there's a lot of communications going on in both of these parts of the central nervous system. And then we need to communicate with the outside and back to the central nervous system. And that's what the peripheral nervous system serves for via these cables that we call nerves. Because if you get pinched on your shoulder, how are you going to know if you don't have a sensory neuron that's capturing that pinch and it's taking it to your brain? So neurons in your brain are processing that, and then it's a pinch. Is the pinch strong enough that I'm gonna, hurt? And what do I do then? Do I scream? Do I, move away from the pinch? So then there's a consequence to that pinch. Like, what is the brain processing for you to do as a consequence of that pinch? So this is just a little example of how things are integrated throughout the central nervous system. Okay, so here's the brain, and it's one of the most magical places in your body, hands down. I'm biased, but it's so true, because so much happens and controls in the brain. So many things are controlled by the brain. We have, so because we're going to be talking about opioids, we have pain signals that are coming up into the brain. So pain signals here, it's good I use the example of a pinch. So you get pinched here somewhere down on your shoulder, out there, right? Out, and because of the peripheral nervous system, you're going to have one of these nerves go into the spinal cord and up into your brain to let you know you're getting pinched. And these pain signals arrive into what we call the thalamus. And we're going to see that again. Thalamus over here. And the thalamus receives these pain signals, and the thalamus has the job to send these signals to different parts of the brain, and mainly, this is one of the main part that it's going to go to. It's going to go to a region in your brain that is involved with sensation, processing sensation. So there are neurons here. See, this is a neuron. waiting to receive information, and it's going to communicate with another neuron over here, and they're going to communicate with each other like, hey, can you believe this? We just received this information from thalamus. We're getting a pinch on the elbow. Is it a, you know, a bee stinging? Is it somebody poking you with a pen? You know, what is it? So we're trying to figure out what the sensation is in that part of your brain. So it's not like a free-for-all in there. Every part of your brain has a specific function, and it's so organized. So and we still don't know everything about the brain, but brain research has really exploded in the last 20 years. Just as a side note, we didn't even know about the adolescent brain 20 years ago, how important the adolescent brain, how important it is to know that it's not as developed as an adult brain. And going to a neuroscience conference a couple years ago before COVID, I attended a lot of the talks about adolescent brains because I had preteens and I just wanted to understand better how they think. And it's incredible when you learn how certain parts of the brain don't develop till you're 25 and don't develop till you're 30, the mature forms of them. And it's incredible because then it gives you perspective on, wow, so they don't have the same judgment, that's for sure, right? Teens don't have the same ability to judge situations like an adult, and that's because their frontal cortex, which is this, and this is the prefrontal cortex, they're not as developed. They're not as developed, so they're going to go do things that might be, you know, more risky. Okay, I'm this is a sidetrack because I find all this so fascinating. Okay, so this slide is really more about to show you how different parts of your brain control different things. So when there is a sensation perceived, right, it comes into the brain and it's kind of interpreted. That's the word I was looking for. Interpreted here in this part of the brain. This is the parietal lobe for anybody who wants to know more specifically. In the parietal lobe. Oops. Then there's a decision that needs to be made, right? That pinch, am I gonna scream about it? Am I just gonna go frown about it? Am I just going to like stay zen because I don't wanna panic? So this is where there's communication between these guys and the motor neurons that are over here. So they are neurons, the cells in the nervous tissue, neurons. Their job is not to interpret whatever sensation you just felt. Their job is to make a decision about, okay, let's fire a command to the muscles of maybe I want to, you know, scream. Let's fire a command to the muscles of your mouth and open your mouth and start your vocal cords, and now you're going to scream because that pinch was hurtful, like, sorry, painful. So these neurons communicate with each other and generate a movement. So yes, in the cerebellum, cerebellum is the little brain behind the cerebrum, right? All of this is the brain. Cerebellum, cerebrum is this entire mushroom cap, and I describe the brain stem as the mushroom stem. The next time you prepare mushroom for your salad, you're going to think of me, right? When you pop that cap off, some people leave the stem because they don't mind it, and some other people pop the stem off because they don't want it, right? So think of it as this is your brain stem, and the mushroom cap is the cerebrum. So this is the cerebrum. And the cerebellum is in the back, and the cerebellum is all about coordination, right? So when you're walking, you're really not thinking about, okay, I'm going to put my foot up and then down and then flex my thigh and then extend my leg. You don't think about all that. That coordination is going to be taken care of by the cerebellum. And all the things that you see This is an eyeball. This is probably a bit too high since, yeah, it's a bit too high here. But the things that you see for vision, they actually, there's of course the optic nerve that's going to capture, the image, but then the processing, I'm not going to draw the line, but the processing actually goes all the way to the back of your brain, and the interpretation is going to take place in the vision center in the occipital lobe. For those who want a little bit more detail here. The occipital lobe. So sometimes, when you fall on the back of your head, you can maybe, well, hopefully it never happened to you, but sometimes people see like a white, because a flash of white, or in cartoons when they have these little stars kind of like popping around. That's because your vision is a bit kind of like, if you fall on the back of your head, there's a disruption to that vision processing. Okay, and one more thing, two more things about this slide is it's showing you memory and reward is actually in very, very specific parts of the brain. This is your hippocampus, and by the end of this talk, lecture, seminar, I'm actually going to show you hippocampus that I dissected out of human brains from opioid overdoses. And these are the regions that I'm studying. The hippocampus is involved in memory formation and memory retrieval, and is very important in addiction, because sometimes remembering a place, or going back into a place that will remind you of a situation, can trigger thoughts of craving. So memory has a very important role in addiction. And then of course, at the basis of addiction is the reward pathway that is being disrupted. So we're going to talk about that reward pathway, and I'm going to talk about these regions in a little bit more detail. Okay, I still have you all on board. I'm looking at all your faces here. Okay, all right. Okay, so let's go. Let's talk about these neurons. So we're really going microscopically now. We're going to look at a cell, the neuron inside the tissue. So you'll have a chance to look at the gross anatomy level. So when we say gross, we're not saying gross like a 5-year-old would say gross. We're saying gross as opposed to microscopic. It's something that you'll visually see. So at the gross anatomy level, the brain, is a structure that you will look at. But at the microscopic level, then you have the cells. So you can't really see them at the naked eye, but you can look at them under the microscope and you will see their cell bodies. You will see them under the microscope. And it's amazing, because it's at that level, microscopic level, that there's all this communication going on. So these cells are called neurons. Neurons. And they're amazing. So they have a cell body where the content is typical of a cell, with all the organelles, the genetic material is in there, nucleus, etc. So this is the cell body, also called the soma. Soma, or cell body. And these cells, instead of just being round cells, like some glandular cells, some cells are tall, some cells are super flat, lining your lungs. The epithelium lining the lungs are very flat, so the oxygen can get through from the air into your body. So there's all kinds of shapes of cells, and they're all very specific for their function. Yep. These cells are stellate, like stars. They're stellate. So I draw them like that, and you probably already caught me do that in the previous slide. So they have a nucleus here, and all kinds of organelles inside. So that's the cell body. However, these stellate cells have a lot of dendrites. Dendrites. So think of them as, these are extensions, cellular extensions, that are very important for the reception of a signal. So at a dendrite, you're going to have another cell that's going to come and communicate with this cell via the dendrite. Sometimes the communication goes all the way to the cell body, but most of the time it's the dendrites that receive the information, right? When two cells communicate with each other, right? They're talking to each other. And then these cells have one axon, usually one, and the axon is where the information goes out. So the information comes in and then goes out and is transferred to another cell. Hey, did you hear Layla got pinched at the shoulder? What? She got pinched at the shoulder. Let me tell, so-and-so, and then so-and-so tells so-and-so, and they talk to each other. And then they interpret that pinch, and then the decision is going to be made about what to do about that pinch. I don't know how I got stuck with the example of a pinch, but here we go. So this is the order of, there's a very specific direction. There's specific direction to the information. So when a stimulus arrives on a cell, it propagates to the cell body, and then it goes back out through the axon. This was the axon from the previous slide. So this is the same cell from the previous slide. You see the cell here? This is the same cell, but now it's communicating to another cell via its axon arriving on the dendrite. So this is the dendrite of the next cell. And then the next cell is going to pass that stimulation to the next cell, right? So the stimulus arrives via the axon of a previous cell, and then it propagates from cell to cell. And when there is a movement, a stimulation for a movement, the last cell is going to be your muscle, right? So at one point, there's we call them motor neurons that are going to communicate with a muscle. Here's your muscle, tendon. And then, this axon is going to go all the way to the muscle and, jolt that muscle with an electrical impulse, and the muscle is going to contract. And that's how your muscles contract. So you can do all these things: flex, extend, move. Okay. Any questions? I can stop, repeat, clarify. Okay. All right. So when you're using a diagram instead of the beautiful pictures before, by the way, just to let you know, this is visualized with fluorescence microscopy. Fluorescence microscopy. So these are fluorescing. This is done in a lab. They're fluorescing so we can see them better. And sometimes we use different colors. Here they're all green, but sometimes we use different colors to actually visualize different things happening. So when we use a diagram to illustrate that, here it is. There's the cell body, and then all of these are the dendrites that are going to receive information, and there's the axon that is going to send the information out. The signal direction goes that way, and then you have a landing on the next cell, just like I described. And the end of the axon is ends as a synaptic terminal. And really, this is where the magic happens. So I'm going to zoom in on a synapse on the next slide. Here we are. So, so this is what we're seeing in this slide. We're seeing this area. This is it. So I'm just going to draw it real quickly. There's something very simple. So this is the end of the axon, and there is like a bulging, it kind of like flares out. And what's inside the end of that axon? anybody knows, maybe we can have some participation so I can hear you, or maybe you know and you want to share your knowledge. You can unmute yourself if you want to tell me what's in here. How are neurons going to communicate with each other? So this is the next neuron.
Those are neurotransmitters.
Yes, very good. Neurotransmitters. These are neurotransmitters. So these are little molecules. This is magic. Little molecules that are going to be released in this space, the synaptic cleft, we call that, because all this is called the synapse. And these are going to be released, and they're going to bind on the second neuron. We call this the postsynaptic neuron, and there's going to be receptors on here. So these vesicles are going to release this neurotransmitter that's going to bind on the receptor on the second cell. So technically speaking, the neurons, the end of one neuron and the dendrite of the next neuron, they actually don't touch. There's a space between them. They don't touch each other. And it's the chemicals that are released between the two that are going to bridge that gap. And the purpose is to create the electrical impulse that came down here, which is called an action potential, if you want to know more details. Action potential. So this is, think of it as an electrical pulse. We need to recreate that on the next neuron, otherwise the next neuron is sleeping. Like, okay, ching, jolted. It wakes up, and now it's going to send that jolt down to the next neuron. So it's an electrical impulse that is recreated on the postsynaptic neuron, the second neuron here, via neurotransmitters. And so these are incredible. Little molecules that are really doing a lot. So this is very simplified here. So these chemical messengers, and you gave me a much better term for that. So I'm just going to say neurotransmitters. That's what it is. So these neurotransmitters are normally inside these vesicles, but if there is an electrical impulse that comes down all the way, this is the axon, remember? This is an axon, and the signal is going down the axon towards the next neuron. So this would be the dendrite of the next neuron. And this one here, this neuron is trying to tell the second neuron, there's a stimulus. You need to send that stimulus, down. And neurotransmitters are going to be released in this space, the synaptic cleft. It means it's that space between the two cells. Okay, so why I'm saying all this, why this whole story, is because we're going to be talking about dopamine when we talk about addiction, we talk about dopamine. So I think it's important to understand what dopamine is. It's a neurotransmitter. So understanding what neurotransmitters are supposed to do in general, like a general function of neurotransmitters, is important. And that's why I took the time to give you that basis of synaptic transmission between two neurons. So hopefully, again, because I don't know all of your background, so hopefully this wasn't either too simplistic or too complicated, that you're all getting at least something out of it. Okay, so neurotransmitters, let's look at a couple examples of neurotransmitters, and dopamine is one of them. When dopamine spikes, it's when we're experiencing something pleasurable, something rewarding. When we get rewarded, we have a surge of dopamine and we feel good about that. And it's in turn, it stimulates seeking for more pleasure. So whatever that initial reward was, it's that dopamine release is going to make us want to go for that rewarding pleasure again. For example, I tell my students, if your professor comes to you and says, you did amazing, your grade on this exam was so much better than last exam, you're really improving, I see that whatever you're doing is working, the studying methods that we discussed together and you implemented are definitely showing a change in the result of your exam taking. So if you get something like that from your professor, acknowledging that you worked hard and your grade improved and acknowledging that progress, you feel good about that. So you're like, wow, this it's true. I got recognized that, I did good on this next exam, and I'm encouraged to do better and to continue doing good, at least for the following exam and by the final. And this is why it's important as professors to always be positive and encourage our students or our children, right, at home. Is, positive reinforcement is really what we want to do, because it has a really big impact on the person receiving that positive reinforcement, because they want to do good. They want to please you again. They feel good about themselves. They want to please themselves again. And they're going to continue studying hard for the next exam and the next exam. So you're seeking for more of that pleasure. And so that's normal. This is all part of life. So dopamine is a good thing. And we have other neurotransmitters that are being released. Serotonin is very important for stabilizing our moods. We feel good. We feel happy, content. And when serotonin levels are down, this is usually in cases of depression, and re-elevating these serotonin levels, bringing them back up is a way to combat depression. And oxytocin, I talk about oxytocin in the chapters where I discuss the female reproductive system and birth and babies and the lactation process. Oxytocin is they call it the love neurotransmitter, because that's what it's a feeling of love, right? It's different from just feeling content and happy, and feeling of love, trust, empathy. These oxytocin release is what gives you these types of feelings. And this is what's released when, for example, a baby is suckling on his mother breastfeeding. During breastfeeding, there's oxytocin levels really high at that time, and there's this bonding that takes place between baby and mother at that time. So it's really amazing. Or every time you give somebody a hug, there's a little bit of oxytocin that is released, and it's supposed to make you feel good. Unless there's other things that come to your mind, and this is not a person you want to get a hug from, and then that's another story. But we're talking about, like, if you're getting a hug from a person you love, this just gives you even more love, feeling of love. Okay, so I'm not sure if you're aware of all of these different types of neurotransmitters. Of course, there's so many different other neurotransmitters. I'm just kind of pointing out to the few that are pertinent to what we're going to talk about, especially dopamine. Okay, let's see, 9:18. Okay. Hopefully I'm not going too fast. But now that you are knowledgeable on the brain, how it works, how neurons communicate with each other, what are neurotransmitters and dopamine, the effect of dopamine, I think you have a good background now to really understand what's going on in the reward pathway itself, and then what happens to this reward pathway when we add opioids in the story. Okay, so when there is a neuron that happens to be a dopamine releasing neuron, so a dopamine releasing neuron is going to release dopamine in the synaptic cleft. That's when you feel a sensation of pleasure and feeling of reward. We just talked about that. And these dopamine releasing neurons, they're not everywhere. I'm going to show you again a picture of the brain and show you that they're called dopaminergic neurons. They're actually in very specific parts of your brain, and they communicate with other parts of your brain, and this is part of the reward system. So, but I'm going to concentrate on now, how is dopamine handled when it's released. So I'm focusing on dopamine. And let me know if you want me to, slow down on something here. So here they are. In the ventral tegmental area. This is, you know what's nice about this session today, this training session, is there is really no exam at the end. Although I will have some verbal quiz just for fun between us, but there's really no quiz, like written exam at the end, so you don't have to worry about missing details or anything like that. So ventral tegmental area sounds like a big word. It's at the top of your brainstem in the midbrain. So the midbrain this is midbrain, pons, medulla, all of this is part of the brainstem. And in the midbrain, there's a subregion called the ventral tegmental area. And this is where these neurons, these dopamine releasing neurons, are located. They're there, they're sitting there until there is a positive reinforcement. So here's, the positive reinforcement is, you hear good job from somebody else. Oh, I'm too much at the edge. You hear good job from somebody else. This is something that, you hear. This is a positive reinforcement that makes you feel good. So this neuron is releasing the dopamine right here. This is where the dopamine is released, in the nucleus accumbens. So if there is one area that you need to remember that is involved in addiction, is this. So maybe you can write this down. This is where things are really going not good when opioids come into the story. But right now, where this is in the normal situation, you have a neuron, a dopaminergic neuron, that is sending a signal to the neurons over here to tell the neurons in the nucleus accumbens, shh, here's the dopamine. This was a really good, we just heard the good job, and the nucleus accumbens goes, whoa, that feels good. This is rewarding. I like to hear good job. I'm gonna, whatever I got the good job for, I'm gonna redo it, and I'm gonna continue working towards another good job. Right? You understand that? So the dopaminergic neuron is talking to the neurons in the nucleus accumbens. Did I tell you this was really important? Yes, nucleus accumbens. And I'll show you dissections of nucleus accumbens and in the human brain when we get to that. So the nucleus accumbens receives all this dopamine, right? It's showered with dopamine. Remember the slide with, let's go back just to show, right here. So imagine this is nucleus accumbens. This is a neuron in nucleus accumbens. We write NAC for short, nucleus accumbens. And this is a neuron that came from the VTA. Far away, right? They're not next to each other. So this guy is in the midbrain, back there, and is sending this long axon to shower this neuron with dopamine. So in the nucleus accumbens, this is where the feeling of reward is triggered. And that is going to go, now these neurons are going to talk to the neurons in the prefrontal cortex. and there's going to be decision made there. Because your whole frontal lobe, this is the frontal lobe. Okay, I wrote that earlier. Frontal lobe, and this is the prefrontal cortex. So the frontal lobe here is your decision making, right? I think I told you that earlier when we talked about movement, sensations, vision. The frontal lobe is your decision making, and this goes back to you know, children and teens, and their frontal lobe is not developed like an adult's frontal lobe. So their decision making is not the same as our decision making as adults. And because a part of the brain that's called the amygdala, and we'll see it again towards the end of the talk, the amygdala kind of regulates fear and interpretation of fear or fearful situations, and their amygdala is definitely not as developed. So they have less fear, and they have you know, poor decision making as far as an adult is concerned. You know, we would find these poor decision making, but in the mind of a teen or even a lower as a child, they're just like, they're not scared, and they're just doing things a bit more with more I'm not going to say courage. It's not about courage. It's about less fearfulness. Okay, so you see how this is all connected? So you feel all this feeling of reward, and then other parts of your brain are going to receive that information as, if this was rewarding, I'm going to do it again. So the decision making here in the prefrontal cortex, the decision making is, I'm going to do it again and again and again. So these guys are going to communicate with the neurons that involve movement, and then you're going to go seek that behavior again. So hopefully it's a good behavior, right? So positive reinforcement for good behavior, and then good behavior will repeat itself. Okay? So this would be movement here. Movement is here, and decision is here. Okay. All right. So all good. So far, so good. And I found this slide. By the way, this is a lot of these slides are coming from the from NIDA, which is National Institute on Drug Abuse. And I like this slide as well, because now this is basically, so I drew it over here. I was, you know, talking and drawing at the same time. And here the pathways are kind of labeled here for you. So what I drew in the previous slide was the dopamine pathway, right? The rewarding pathway, the reward pathway. And I was talking about VTA, right? That's in blue here. Nucleus accumbens, remember, I told you this is really important over here. And then from here, we actually have a communication with other, even from nucleus accumbens, even though it's not shown here. And then quickly, I can show you the serotonin pathways, because in case you're interested also about depression. Serotonin pathways, it stems from just a slightly different region in the brainstem, the raphe-nucleus, and there's going to be communication with all different parts of your brain. And like as I mentioned earlier, serotonin is more involved in mood, memory, processing, sleep, and cognition. So lower serotonin level usually leads to depression. Okay, I need to have a sip. Okay. So this is the you know, nicer diagram from NIDA. which is a great governmental organization that has so much information about different drugs and that. So those are the vesicles that I was talking about. Vesicles. Filled with dopamine. So there's only a drawing of 3, but of course there's so many. And then eventually this dopamine is going to be released. And how does the dopamine act on the second neuron, right? This is your next neuron. This is the second neuron. Or more appropriately, we would say this postsynaptic neuron. Presynaptic neuron is this one because it's before the synapse. This is the synapse, and this is the postsynaptic neuron. So there's a receptor here. A receptor on the postsynaptic neuron in the membrane of the postsynaptic neuron, right? At the dendrite level, we talked about that. And I'm sure you probably all have at least a basic biology background, and you can kind of identify this. What do you recognize this from, you know, a biology course you took in the past? What does this represent? This is more detailed than just a line. Do you know what this is?
A cell membrane?
Yes, that's the cell membrane, exactly. These are phospholipids with the phospholipid heads and the phospholipid tails, and they're facing each other, and they're creating this very hydrophobic, lipid-rich layer. And this is basically what's at the edge of a membrane, at the edge of a cell, is this cell membrane. Why I'm saying that is because all of these are lipids, so it's a lipid, we call it a lipid bilayer. But inside this lipid bilayer, you have these proteins. Receptors are a type of protein. And we have all kinds of proteins in the body doing all different things. You know, some proteins are just meant for structure, like collagen. Collagen in bone and cartilage. This is just a protein for structure. And other proteins are for accepting a molecule, like a neurotransmitter. So this is a receptor. The molecule itself is a protein. The job is being a receptor. So it sits there. You know, think of it as, you know, you're sitting here and you're looking upwards and you have your hand cupped like that, and you're like, oh, is it going to be today? Am I going to get dopamine today, right? And you're just waiting and waiting and waiting until dopamine is released, and then it's captured by the receptor, and then you have the feeling of, yes, it's today. She said good job. She said good job. Professor Jabour said good job, so I'm good. So these receptors are there to capture the dopamine released by the vesicles. So when the dopamine is released, here we go. The dopamine, which was illustrated with this big, chunky shape, sits on the receptor. But which is really important, there's an uptake pump. So this is another protein. This is an example of another function for another protein. So this is also a protein, but its function is to pump the dopamine back into the cell, to recycle it. And also, can you tell me why do you think pumping dopamine back into the cell is important?
So dopamine doesn't get depleted?
So dopamine doesn't get depleted, correct? That's recycling it. So we have dopamine again in the presynaptic neuron, correct? And what else? What's the other consequence of the uptake of dopamine?
It limits how much dopamine is in the synapse?
Correct. So for these two reasons, and the the biggest reason, well, they're both important, is to not leave dopamine in the synaptic cleft, in the synaptic space at the synapse. The whole region is called the synapse. We do not want the dopamine to stay there because we don't want this to be prolonged. In a normal situation, we have dopamine recycled and it's gone. And it's like, okay, and then you move on with your life and you go do other things. And then the next time you hear, good feedback from somebody, then there's another dopamine release and then another good feeling, and then that's recycled, right? So it's intermittent, it's not prolonged, and that's how we want it. We want it to be recycled and out of the synaptic cleft. So that's the important part. And you'll see how drugs make the dopamine stay in the synaptic cleft. That's how the drugs are tricking the synaptic transmissions. So I want to mention before I move on to the effects of the opioids on all of this normal functioning here, is we also have modulators, neuromodulators. So endorphins are one type, neuromodulators that can be provided by other cells. So this is actually here, it's another cell that releases this neuromodulator called endorphin, and the endorphin binds on the opiate receptor on this postsynaptic neuron. So this postsynaptic neuron has, the dopamine receptor, but also has an opiate receptor, and the opiate receptor will bind to the endorphin, and the endorphin basically enhances that feeling of the good feeling. So there's the reward feeling and the feeling of feeling good about that reward, that feeling of reward. So it's together. So it enhances the response. So this is a neuromodulator that actually enhances that response from dopamine. I'm going to talk about another neuromodulator in a minute. Okay, so natural rewards. So these are the 4 basic natural rewards. So my example didn't fall in one of these, but because my example was, hearing good job from a professor to a student. But the 4 natural rewards that we have in life is food, water, nurturing, and love. So relationships. We go after that. If it makes us feel good, we go after it. We need it. We seek it. Yeah. So now that we understand how things work in a normal scenario, and it's 9:37, okay. Now that we know how things work in a normal scenario, and I think if I'm not going too fast, and you can tell me to clarify anything before we get into the opioids, I think you have a good understanding of how neurons communicate, how we have neurotransmitters that are released from one cell to the next, and how these neurotransmitters, depending on what they are, in the case of dopamine, we end up with a feeling of reward, a good feeling of reward. Okay? So let's see how this can be really disrupted with the use of opioids. Any questions so far? So I like to start with a couple definitions so we can understand the different terminology. When we talk about addiction, it's really a state, and this is applied to so many different things, not just drugs, a state in which an organism engages in compulsive behavior. That's the word here, compulsive. You can't stop yourself from doing that behavior. And there are so many different types of addictions. Of course, we think of the drugs, the different types of drugs. We think of alcohol, we think of nicotine addiction, sex addiction, addiction to gambling, and I even have a personal story of a cousin of mine who was addicted to running, where she really, she couldn't go without running, and she did it so much it disrupted her her menstrual cycle, and it delayed her being able to have children because of that addiction. So there is such a thing as being addicted to into a physical activity. When it becomes detrimental to your health, that's when you know that you've passed the line of, I just love to do it versus I can't stop myself from doing it and it's detrimental to me. So this really, that there's that line there. So the behavior is reinforcing, meaning it's rewarding and pleasurable. So of course, in the mind of these individuals, they feel good about doing it, so they keep doing it, but they have lost the control of limiting how much they do it. So it's okay to feel good about something and doing it over and over, but if you can't spend a whole weekend without doing something because you're now drawn into doing something else for family reasons, or you can't do your initial that behavior that you don't want to stop doing, and you can't actually do other things, and that's where the problem comes into play. So I wanted to show you this slide because this is actually a slide to explain how the discovery of the reward pathway, how it was established. We use the rodent model in research, mice and rats, and by using these animals, we're able to understand what's going on in the brain. So it's very, very helpful for us to progress in the world of research when we're using these animals. Of course, every time there is animal use in our research, we have to go through a very stringent process in the application to show we're using ethical methods and all of that. So there's a committee that just judges the ethical aspect of your research even before you start doing that research. But the discovery of this reward pathway was achieved with the help of these animals. And so the picture I'm showing here is the representation of an electrode that was placed in the nucleus accumbens of rats, and when the electrodes, the rats had access to a lever that they could press, and it would send a jolt to the nucleus accumbens. Can you imagine the setup, right? And I'll show. I think I have a picture a little bit later about that. So the rat basically administers itself a jolt to the nucleus accumbens, and we discovered through this kind of research that the rats keep pressing the lever, and when we're talking about the jolt, we're not talking about like a taser or something. We're just that little tiny tiny electrical impulse in the nucleus accumbens, and they like it, and they press the lever, they press the lever, they press the lever. If we move the electrode to a different region outside of the nucleus accumbens, they just like walk around. They don't go back to that lever because it's like, what is this? It doesn't do anything, or it doesn't stimulate anything. So this is how we discovered nucleus accumbens is really at the center of all the story. This is where the dopamine is released. So the neurons that release dopamine, they may be somewhere else, right? So in this diagram, they're here in the VTA. This is where they're sit, the cell body of these neurons, but then they send these long axons and shower the neurons that are sitting in nucleus accumbens, they shower them with dopamine. So the the jolt here is equivalent to a neuron in the VTA that's going to communicate with it's basically sending the electrical impulse that normally would have come from the VTA. This is now being administered artificially. And this is really how we discovered the importance of these regions. So we use that, and we understood that when opiates were involved, when opiates were added, were given, they basically they travel to the brain, and they have very specific receptors in different parts of the brain. So what's highlighted here is the reward pathway, which now I talked about it a couple times, right? VTA and nucleus accumbens right here. This is the reward pathway. However, opiates also have an effect on the thalamus. This is the thalamus. They also have an effect on another part of the striatum, the caudate. And then, you know, and so many different other parts of the cerebral cortex up here. So there's actually an action of opiates other than the reward pathway, in addition to the reward pathway. And we're going to also talk about that. So I'm introducing one more thing that to your tool of knowledge about dopamine and how it's regulated. So far, what I told you, and you seem to have understood that when dopamine is released, let me see here. Yes, so these are my little dopamines right here. Dopamine is released over here from the vesicle, remember? Dopamine is released and binds on the dopamine receptor, and then you have a response in the postsynaptic neuron. This is the membrane of the postsynaptic neuron. Okay, so and we talked about the recycling of dopamine and how, you know, dopamine can be recycled. However, there's a way to modulate the release itself, so we don't have too much dopamine in the synaptic cleft, right? The synaptic space here. We also don't want too much of it released. So there is, this is natural, this is normal, there is a GABA. This is called GABA. GABA is another example of neuromodulator. Do you remember when I talked about neuromodulator? And GABA is released here as a neuromodulator. What is the other neuromodulator I talked about earlier? Do you remember?
Endorphin?
Yes, excellent, very good. Endorphins. So endorphins are released, but also GABA is released. GABA is important in GABA is important in inhibiting too much release of dopamine, and I describe it as, I give it the equivalent of like a parent setting a curfew. Okay, you can come out, but you know, not too much. That's what it means. It's not too much. So there is a negative inhibition of the, we call it exocytosis, the release of dopamine into the synapse, the synaptic space. So GABA is really important in regulating the release of dopamine. So this is great. This is in a normal situation. Yeah, I forgot that I had all these circles for you. Now, when you add opioids into this story, when you add opioids, it modulates the release of GABA. Oh, bummer. So let's see where are we here. This is heroin, and heroin is metabolized. When you take heroin, you actually, it gets metabolized into morphine and another byproduct, and then these byproducts the products of this metabolism, it binds to the opioid receptors. We talked about that on the cell. So at the time, the opioid receptor was on the same cell as the dopamine receptor, and this opioid receptor is on this other cell. It's in the vicinity. It's on either cells. And what opioid receptors with a heroin byproduct binding on this opioid receptor. So the activation of the opioid receptor, what it does is that it inhibits the release of GABA. That means we have less GABA modulating the release of dopamine. I know it's like a inhibition of an inhibition. So it's like, okay, let's make sure we have that clear, right? It's not a direct way of stopping things, but it's basically inhibiting the inhibitor. No more curfew. Dopamine is released in higher quantities. So this is the problem for opioids and their receptors. When their receptors are activated, they modulate the release of GABA, which in normal circumstances, it modulates the release of dopamine. But if we don't have a modulator, then dopamine is released like uncontrollably and in high quantities. And now we're really bathing in, ooh, this really felt so good, right? So it's like an even higher feeling of reward. So this is how opioids work. Other drugs work in a different way for the same final outcome, more dopamine in the synaptic cleft. Could you give me an example of how we can have more dopamines in the synaptic cleft otherwise? I think we talked about that earlier. We talked about how to prevent having too much dopamine in the synaptic cleft. What would that be? Another way that we would influence having too much dopamine in the synaptic space here.
The uptake pump.
Exactly. This uptake, this transporter, this protein at the membrane that is supposed to recycle dopamine. In the example of cocaine, the cocaine acts on this uptake molecule, and cocaine inhibits the recycling of dopamine, and that's how you end up with more dopamine in the cleft. So I just want to give you that side example of cocaine, even though I don't talk about cocaine in the rest of this presentation, is to show you that different drugs do different things, but in the end, for at least cocaine and opioids, the end result is we have this dopamine release that is higher than normal. So that's what it is. So just as rats were stimulated themselves with a small electrical jolt, and that's how we discovered the reward pathway and the importance of nucleus accumbens. If you do the same with a source of heroin, the rat is going to go to that lever over and over and over again. So they will administer themselves, self-administering heroin through this small needle that's placed into their brain and directly into the nucleus accumbens. We also do that with a needle outside, so in their system instead. It just takes it takes longer to get to the brain. So there's two approaches to these types of research. So scientists have found that dopamine is increased within the reward pathway in the rats that self-administer heroin. So they tied the two together because we didn't know, we didn't know the role of dopamine. We didn't know the reward pathway to begin with. So all of this took research to really pinpoint, figure out. Not only do we now understand the role of nucleus accumbens, but we also understand the consequence of the release of dopamine in those regions. in this region. There's really many videos online. I just have one here from pbs.org, but you know, depending on your knowledge level, you can go find videos that really talk about the opioid effect in even more details. So this is the big picture. Just kind of like wrapping up what we said, the big picture is that opioids will bind to opioid receptors in the nucleus accumbens. And when there is and it has the effect of a rewarding feeling. So the dopaminergic neurons are here in the VTA, and then they send signals to neurons in the nucleus accumbens, and then you have that feeling with the higher dopamine release, and this will be, you know, transmitted to other parts